We have heard a lot about CRISPR, how it has revolutionized genome engineering, leading to widespread applications in agriculture and human health. But I'm not here to talk about genome engineering at all, because CRISPR has many more tricks up its sleeve than just genome engineering. I'm going to talk about biosensing. The application that scores of people use in everyday life to know if they're pregnant, or to check for their blood glucose levels, or of course to know whether you have contracted the SARS-CoV-2 coronavirus. That biosensing. A biosensor, simplistically speaking, needs two crucial elements: a target binding part and a signal generation part. So essentially, once our molecule binds with the specific target, it needs to generate a signal allowing us to correlate back the specific concentration of the target we are trying to detect. Now, CRISPR has jazzed up and left its mark on the field of biosensing as well. And that's what I'm going to share with you today. To give you a quick look, I'll share the work of Biosensors Group here at KU Leuven on CRISPR-Cas9 based biosensing. And I will also introduce two other very promising CRISPR tools that have been explored for biosensing: the CRISPR-Cas12 complex and the CRISPR-Cas13 complex. So let's begin. So we know what a typical CRISPR-Cas9 complex looks like. We have the CRISPR RNA, the tracer RNA, and then the Cas9 protein. Now, if we use a dead version of Cas9, which is essentially catalytically inactive, then this complex functions a bit differently. If the dead Cas9 is used together with CRISPR RNA and tracer RNA, then the complex still searches for a DNA target and it still binds with the DNA target. However, there is no cleavage of the said DNA target. So essentially, this works just like a DNA binding molecule, which means that half of the function of a biosensor is done. Now, what about the other half? The signal generation half of a biosensor. If we couple, for example, a green fluorescent protein with the CRISPR-Cas complex, then that can give us the signal generation part. This is just one example. But as you see over here, if we do couple such a signal generating moiety with the native traditional CRISPR-Cas9 complex, we can generate signal upon binding of the CRISPR complex with its target. As you see here, this is precisely the technology that has been used for cell imaging, for instance, for imaging of telomeres in live cells and other similar applications. We at the Biosensors Group at KU Leuven have engineered another way of signal generation. We have engineered tracer RNA to include a DNA sequence, making it a tracer RNA DNA hybrid. Now, this DNA can function in two distinct ways. It can work as an enzyme on its own, which is actually a DNA-based enzyme or DNAzyme, such that this DNAzyme, once it cleaves its specific substrate, can generate a signal. Or it can serve as a site for hybridization of a labeled DNA probe, which enables downstream signal generation by coupling with other enzymes. This way, we achieve an amplified signal generation, which enables even more sensitive biosensing applications. Now, we have used this engineered complex in 3 distinct settings for potential DNA detection applications. Firstly, on membranes. Here, the target DNA is spotted on a membrane and then enters our engineered CRISPR complex, this time with the DNA-zyme sequence included in it. After we wash off the excess, we incubate this membrane with the DNA-zyme substrate. Now, this DNA-zyme substrate is labeled with a fluorophore and a quencher on each end. So once DNA-zyme cleaves the substrate, there is an increase in fluorescence. That's what we have on the right side. Fluorescence signal in the presence of our target plasmid and none in the presence of a control plasmid. We also use this same complex, the one with DNA-zyme domain included in CRISPR-Cas9 complex, for detection of DNA target on microbeads. Here, instead of a membrane, the DNA target is captured on microbeads, and then the engineered CRISPR complex is introduced, which binds this DNA target. After the excess is washed off, again, we introduce the DNA-zyme substrate. And cleavage of this DNA-zyme substrate again results in an increased signal generation, which is concurrent with the increase in target concentration, as we see over here, leading to detection of the DNA target in nanomolar range. In another instance, we have used the alternative CRISPR DNA approach, where the CRISPR-Cas9 complex has a DNA domain added instead of a DNA-zyme domain, to again detect DNA target captured on microbeads. Here, the DNA probe hybridizes with the DNA part of tracer RNA DNA hybrid, and it has a biotin moiety attached to one end. We use streptavidin conjugated beta-galactosidase enzyme for signal generation, which binds with this probe via biotin streptavidin binding. This complex is then seeded onto our in-house developed microwell array platform, wherein each microwell holds a single microbead, and we can quantify the amount of target molecules in a more sensitive way. Using this combination of bead-based assays, engineered CRISPR complex, and microwells, we can increase the sensitivity of our bioassay, detecting targets in the femtomolar range. As you can see over here, the microwells with target molecule, and hence the CRISPR complex and the enzyme, literally light up, allowing us to count the presence of target molecules in our sample. And there is a lot more cooking from where that came from. There are other strategies also being explored for CRISPR-Cas9 based biosensing, which only point towards the promise that this technology holds. But the story of CRISPR does not end with Cas9. CRISPR is a much more diverse tool, and it has a lot more tricks up its sleeve. I'd like to share just 2 more examples of CRISPR complexes that have been used in biosensing to give you an idea of the power of this revolutionizing tool. First up is Sherlock. Now, this biosensor utilizes the CRISPR-Cas13 complex, which is different from the Cas9 complex we've seen earlier in 2 distinct ways. First up, it binds to single-stranded RNA as target. And secondly, it has collateral cleavage activity once it's activated by binding with the single-stranded RNA, which means after binding with the target RNA, it will indiscriminately cleave single-stranded RNA in its vicinity. In this biosensing strategy, hence, the collateral cleavage is made use of for signal generation. The reaction mixture has fluorophore quencher labeled RNA molecules so that they are cleaved via collateral cleavage and result in an increase in fluorescent signal. So where does the RNA target come from? In these biosensors, there is upstream processing of the target nucleic acids. For instance, DNA target undergo isothermal amplification to generate exponential number of copies, which are then transcribed back into RNA. Alternatively, the RNA targets are converted to DNA, similarly amplified, before being transcribed back to RNA. Hence, the sample processing is a pivotal component which is required upstream of this bioassay strategy. Using this strategy, detection of cancers, viruses, single nucleotide polymorphisms, and even the SARS-CoV-2 virus, which causes COVID-19 disease, has been shown. And this technology is further being developed for commercialization by Sherlock Biosciences. Another key player is detector. This one uses CRISPR-Cas12 complex, which is similar to Cas13, except that it is activated by binding with DNA target. And also, it cleaves all single-stranded DNA in its vicinity after activation. So similarly here, the biosensor uses fluorophore quencher labeled DNA molecules which get cleaved, resulting in an increase in fluorescence. In this case as well, similarly, the DNA target is amplified before being added to the reaction mixture for analysis and quantification of the target molecules. This technology has been used for cancer screening. It has also been shown to work for detection of bacterial infections, antibiotic resistance, and also for detection of the SARS-CoV-2 virus causing the COVID-19 disease. And also, this technology is being further developed for commercialization by Mammoth Biosciences. We believe this is only the tip of the iceberg, and this revolutionary technology has a lot more in store for delivering innovative CRISPR biosensors. With this, I leave you today. We'll continue exploring this nifty tool and invite you to stay in the loop by connecting with us here at the Biosensors Group at KU Leuven. Thank you.